So, in picking a topic for this presentation, I thought about how funny it is that our elementary school science experiments are so revolutionary to people 150 years ago. For example, take tonic water. If you shine a black light through tonic water, it's going to glow a bright blue. And we know this happens because a chemical reacts with UV light to make this blue light. But in 1845, scientists were kind of left scratching their heads. That is, until Sir George Stokes coined the term fluorescence, which is what I'll be presenting on today. To explain it a little better, take a look at this energy diagram here. These arrows represent electrons, and the lines represent energy that the electron has. Higher line means higher energy, and vice versa. But our electron starts here, at the bottom, which we call S0. Light is going to hit it this way, and as it gains energy from that light, it gets excited. It's like bouncing on the trampoline, and then all of a sudden someone else bounces at the same time as you. You get where I'm going with this. So the electron flies way up to this very high energy state. But this energy state it goes to is unstable, and it can't stay there very long. As it falls down, it emits a particle of light. That particle is what we measure because that's what fluorescence is, and we have to measure it because that electron inevitably loses energy. So the emission wavelength is going to be longer than the excitation wavelength, and that's called the Stokes shift after that guy Stokes who, discovered fluorescence. But our goal is to get that ratio known as the quantum yield as close to 1 as possible. So what type of fluorescent molecules do we have? I'll mainly focus on two. There's fluorescent proteins and fluorescent polymers. Fluorescent proteins were derived from green fluorescent protein, which is actually found in jellyfish, and those have been the building blocks for everything since then. Because every protein is just a sequence of smaller building blocks called amino acids, scientists found that you can actually change the order of these amino acids to change the color of your dye. That's pretty cool, right? And as far as fluorescent polymers go, the most important word to look for is conjugation. In covalent bonds, electrons are shared, like a family-style dinner among everybody at the table. Conjugation means that your molecule has alternating single or double bonds. And typically, these types of bonds are very reactive, so they can be attached to a loose end on a molecule and make the bond light up. Okay, so this all might seem pretty overwhelming, right? But don't worry, because selecting a dye has never been easier thanks to the magic of the internet. All the big manufacturers of fluorophores all have sheets attached to every popular dye they sell that tells you exactly what the statistics of your dye are. And you can even get clues in the name itself. For example, take Alexafluor 488. The name for this dye implies that it can be excited by, you guessed it, a 488 nanometer laser. Instruments in fluorescence analysis of labeled particles are used to figure out just how much fluorescence we have in a specific area to look at what exactly is lighting up. Generally, fluorescence instruments kind of look the same. First, a laser shoots at the particle and it passes through a prism, which separates the random light into specific wavelengths. You might have seen this before on a specific album cover that I probably don't need to name. But those lights go and hit your molecule, and the molecule releases light itself, just like in the energy diagram from earlier. What's unique about this light is that we have to read the photon at a 90-degree angle. And we measure it there so that our laser doesn't mess with our outcome. We don't want to lose our sample in all of the scattered background light. So that light is filtered then, so we only get what it releases and not any extra background. And it's read out by a detector, which is usually a computer program. And what you'll notice is if you study this long enough is the only thing that really changes between instruments is the way that your light is actually read. For example, a microscope is pretty similar to this setup, only you need a lens and you need mirrors to flip the image like a camera so that your eyes can actually see what you're looking at. How do we apply all of this then? So applying what we learned, these proteins and polymers can help us to actually build a structure of the cells in our bodies. If we label the nucleic acids in our DNA, for example, or components of our chromosomes with the green fluorescent protein from earlier, we can actually see how DNA and RNA move around in our cells. And if we label them with two different proteins at the same time, we can actually see both at the same time. Additionally, we can use labeling to identify antibodies. We can identify structural components of our cells, like the extracellular matrix. And we can also look at different fats in the body and where they're stored. Obviously, there's very far to go in this field. We've already come so far, but there's so much we have to know. But from this, it should be clear why fluorescence labeling has this longevity, and it's so important to be able to tell what's going on in our body. Quite frankly, fluorescence labeling is the basis of all modern biophysics. And I don't think we'd be able to actually look at anything without it.